Look at that, Trevor, so on it. Got it. All right, hi everyone. Today I'm going to be introducing our seminar speaker, Dr. Matthew Eby, who is currently the chief scientist at Booz Allen Hamilton, and he focuses on working with federal clients. Before that, he was a program manager at Universal Technology Corporation, specializing on working with the Air Force, Navy, and Army. He did 2 postdocs, one here at UGA and one at Oak Ridge National Laboratory. And he got his PhD in microbiology right here at UGA after his undergraduate degree at Rutgers in environmental science management. And today he'll be talking to us about bioinformatics consulting. So let's welcome Dr. Eby.
Thank you for having me. Thank you for the invite to I'll double check. A message saying nobody. Oh, okay, cool. All right, cool. Just glad everybody can hear on Zoom. Yeah, so my name, I go by Matt, chief scientist with Booz Allen Hamilton, and appreciate to talk to you a little bit today about bioinformatics consulting. So this isn't really in chronological order. We're just I want to touch, other than the introduction, but I want to touch on a number of different things here. Want to give you some examples of some bioinformatics consulting that we do within Booz Allen, and also with some other projects. Talk a little bit about bioinformatics consulting career track. How bioinformatics plays into the larger creation of solutions to tackle complex problems that we see within the federal government. I think mostly what I'm going to talk today, and the examples are going to be federal government bioinformatics consulting, and also giving you some examples on bioinformatics as it pertains to genomics. And also with public health. But I'm going to try to make sure that we talk a little bit about bioinformatics consulting that I feel could be applied to outside the federal space, as well as necessarily with public health. And really what we're going to try to hone in here is that with bioinformatics consulting, the bioinformatician is at a very central, critical piece to a larger team that works solutions in consulting, but it's a team effort. So bioinformaticians work with a lot of other skill sets in order to get some of the most difficult problems tackled into solutions. Just quick about myself. Thank you for the introduction, but I'm going to go down here my little pictograph of my my life trajectory. I grew up, went to high school in rural Pennsylvania, and at the first chance just wanted to get out of rural Pennsylvania. That led me to a undergraduate degree in environmental science at Rutgers. This was right out of New York City, so in northern New Jersey, so definitely got out of rural Pennsylvania. Got my my bachelor's there, learned about a program at the University of Georgia at the time, which provided an NSF research grant to help pay for my schooling, as well as give me a stipend. Tremendously successful and awesome program. So I jumped at that. Went into the microbiology department with Dr. Ellen Neidl. And there, really, between Rutgers and environmental science and going into biodegradation and catabolic pathways of aromatic compounds, which directly relates to, in a lot of cases, bioremediation, really had this idea that I'm going to have some of the world's worst polluters pay me to help clean up their mess, right? After that, I had an opportunity either to do a postdoc, was in consideration at Yale, but ultimately chose to go through what was called an ORISE fellowship. If people don't know what ORISE fellowship is, it's Oak Ridge Institute of, I think, Science, Education, or Engineering. Anyways, Oak Ridge National Lab has fellowships for graduate students, for postdocs, for even professors to do kind of like 2 and 3 year fellowships at certain institutions. A lot of times they're federal institutions. I can go into a little bit this later. I can also talk to students about how this actually brought me to the Air Force Research Labs. I did a postdoc there. I'm doing bioremediation of explosive contaminated waste sites. At that point, I finished my fellowship. They wanted me to continue to do my work, but at that time, there was a broad hiring freeze with the federal government. And so the mechanism in which I could still continue to do my research, but didn't have the opportunity to become a federal employee, was to become a federal contractor. So a company picked me up, paid my salary. The next day I came in as a federal contractor to continue my job. Spent 10 years with Air Force Research Laboratory, eventually as a senior scientist to a program manager, and then ultimately about 10 years ago, moved back to Atlanta and got a position with Booz Allen, where my major client is CDC and other health and human services agencies such as FDA and NIH. So either that trajectory, just looking, it looked like that either that was the trajectory that happened for me. That's how I made it into federal contracting and consulting, right? So, and I'm certainly willing to answer questions more about, how you kind of make it into that track. I think my track is a good example that's still relevant today. And I can talk a little bit more of that. It was either that or it was just basically I only worked for institutions that had red in their color. I don't know.
It's a good call.
Okay, so super quick, this is the advertisement. If you go Google Booz Allen, you're going to see we're a 35,000 employee company. You're going to see that we're management and IT consultants. But what is changing with both the consulting firm that I'm working with, as well as many consulting firms, is that they're putting a lot of investment, time and energy in going into what's called life science consulting, right? It is a field of science that they see a lot of white space, meaning there's a lot of opportunities and funding to move into that. One of the two major thrusts that are going on, particularly at CDC and other federal institutions, are two things: is data modernization, right? So the administration has dumped billions of dollars into federal agencies to modernize their IT infrastructure, right? And part of that, particularly with the agencies that are part of the health and human services, that's going to be the cross-section of IT modernization and life sciences, right? FDA. The other part of that is because of the pandemic. So a couple of years ago, there's tons of money that went into the pandemic. That money is going to continue. Unfortunately, I don't think we're going to be anywhere near done with future pandemics. And so there is this intersection of public health, largely life science consulting, and IT modernization, that they are looking for a lot of skill sets to hire and to pay good money for that. And talking about those keywords, I mean, what else is bioinformatics, right? Is within that cross section. So I just, go ahead and Google, if you want to learn more about Booz Allen Life Sciences, just go ahead and Google it and you're going to come to our page and there's lots of examples. All right. There are three main components in the larger of what I just talked about data modernization of federal agencies that do life science technology, right? The three things: there's researchers, right? Those are the people that generate the data, right? They have to put the data somewhere, right? So you have researchers generating data. In the part, there's quality assurance, there's lab safety, there's a number of associated processes and skill sets and jobs that go along with the researchers. They have to put the data somewhere, right? So database administration, the applications that are associated with database are built by data architects. They're used by data scientists. They're built by IT developers. And all of that is on our third point here, which is the technology around that, okay? And you can see in the center here, you know, this one, an example is biomedical informatics. But as I said before, about bioinformaticians being kind of a key linchpin to all of this is part of this, right? Because assuming the bioinformatics graduate student, you're generating data in a lot of cases. I think I heard a few people were not really happy about the wet side that they had to do for their dissertation and stuff, but it's being done, right? So, and that means you're generating the data, but then you're building the bioinformatic pipelines, and so you're using the technology, right? So that's a great place to be in the middle of where a lot of funding and a lot of innovation is being driven through life sciences. All right. The most complex and difficult solutions are multidisciplinary solutions, right? So, and this is a cross-section, you know, grouping those people together that form a team to a lot of consulting that we do for the federal government. You know, once again, starting from the top left, we have the life scientists that generate a lot of the data. Epidemiologists and statisticians are the groups that relate if that data has a human element to it or is relevant to a human problem. You have the epidemiologists and statisticians that crunch those numbers with respect to an issue or a problem or a project that deals with public health. You know, grouped close to that are the data scientists and the bioinformaticians also, right? So you do very similar processes to the epidemiologists and the statisticians, but a lot more data intensive in a lot of cases, data heavy, and a lot of the innovation that is occurring there. Associated to that, which we're going to talk about a little bit, because we do have some teams and projects based on quality management. You know, a deployable public health bioinformatics system has to be robust, it has to be defensible, it has to be validated, and that is where is a very key component in quality assurance that has to be done. You know, it's one thing that if you're doing research, you know, at an academic institution as maybe part of your dissertation and whatnot, it's different when you are trying to discover where the next COVID variant is going to show up, right? Technology experts, this is the group, because, you know, you need the database administrators, you need the developers, you need the data architects, all of that heavy IT side, of course, that builds these platforms is part of those technology experts. And then you have people like myself or the management professionals that kind of run the team, you know. Honestly, probably the least important person of the group here, but is still needed in order to keep the team cohesive and do things like the project management and in particular with IT development, you have to do IT development in a very specific way and manage it in a specific way for high efficiency, for low, you know, as low cost as possible, and to get those deployed assays or pipelines or platforms out there that are going to work in the public health field. All right, I'm going to go through couple examples of what we do currently. This first example is part of what is a very large genomic database that's being formed called the Millions Vets program. Pretty self-explanatory. Basically what they're doing is they're collecting samples, genomic samples from up to 1 million veterans and putting that into the database. That database, it's very, very similar to 23andMe, you know, Ancestry in some respects, is to get that large data set where you can start looking at association genotypes. You can look at the epidemiological phenotypes and try to connect those and make connections in order to really focus on and understand things like PTSD or suicide risk or cardiovascular. Some hearing loss is 1 in 5 veterans have hearing loss. It's a multi-billion-dollar expense for the government to deal with our veterans who have hearing loss. There are genomic associations based on hearing loss, right? So if you can take a look at a group of subset of these veterans and determine genomic determinants of hearing loss, then how can that help inform the military in reducing the costs, the healthcare costs of hearing loss? So for example, you know, if you're going to be predisposed for hearing loss, maybe you're going to be required to wear hearing protection a lot more than maybe another veteran or another military person. Within these surveys, there are Gulf War surveys, there have been COVID-19 collections, and then also minority-specific arrays that have been used as part of this system to help flesh out those nuances between genomic determinants and disorders and disease as part of the military. That leads to what's known as precision medicine, right? So precision medicine is you take those genomic and phenotypic determinants, you take all of that data together, you find those associations, and then you apply personalized precision medicine to those groups. So one example of the hearing loss, right, is you know, that would to help lower the incidence of hearing loss. You know, there may be environmental factors get put into play where you have people wearing extra hearing protection. The same way with, you know, cardiovascular disease, or you know, having the health monitored by the military over time to ensure that it doesn't lead to major health problems when they become veterans and later in life. Okay, that leads to certain concerns that the bioinformatician needs to understand as they do their job, right? Now, you know, I showed you that team slide before. You know, there are quality specialists that take a look at the quality. There's data specialists, there's data security. Those people are those have specific jobs that come part of that team to make sure that you get a data set that's high quality, that's secure, and is robust. But understanding, knowing that the bioinformatician isn't generally usually at the center core of that data set. They're the ones that's massaging, manipulating the data, grouping it together, determining, building the pipelines to find those associations. The more you understand about these concerns about the data, the better you'll fare in the workplace to be successful, all right? Obviously, data quality, we talked about that, security too. PII, PHI, right? Personal identifiable information, personal health information. This is highly controlled by federal institutions. They can only be, you know, this type of data can only be stored in very specific circumstances. A lot of times, if, you know, you as a bioinformatician need to view these data sets and make the associations as pictured here, there's ways of de-identifying the data, right? Removing the patient name, removing all of that so that it's de-identified, and it can be used then in a less secure environment for you to do your bioinformatic processes and pipelines. That goes to data access, right? Depending on your access level to this data, whether you're allowed to, you know, just like how in the military there are security clearances like top secret and secret, there are similar data access restrictions as part of with health, having what's called public trust clearances. Dual use, right? Dual use means that while you are collecting this data for doing something good, could somebody use that data to do something bad with it, right? And in the military, that's something that's very, we're trying to address and is being very closely looked at, because if you get all of this deep genomic data from our military population, our veterans, and you determine that, this group of people have an issue that could lead to increased healthcare costs by the military, easy thing would be to do is just not let them into the military, right? That's pretty fair. So there's ethical solutions, there's bioethical solutions, are concerns and issues that you need to work with this. And then also acceptable use, right? When you collect this data, okay, let's say you go to, the doctor and they take blood to get your white blood count, right? You went into that doctor's office with the understanding that the test that was done on your blood was for a white blood count, right? If we store that sample away, a couple years later, and there's a medical institution that has a number of those samples, they want to start looking at how many of those people had HIV or hepatitis, right? Is that fair to the patient? Would you really want your unknowingly that somebody is using your test and your blood sample for things other than you agreed to do, right? And so acceptable use is a big issue. It's great to collect. It takes a lot of time and energy to collect these samples. There's 1 million veterans, there'll be blood samples that are stored in cryogenic freezers that could be used in the future, but you can only use those unless it was expressly approved and understood by the patient itself. So as a bioinformatician, it's really not your primary job to think about these things. But if you do think about these things while you're collecting this data, you're going to you're going to be much more desirable in the workplace, because when you create these data sets and you're thinking about these, then these data sets are ready to be used for the future because you address these different issues. So, in these examples, this is where, as a bioinformatician, in collection of that data, there's going to be platforms that are going to be built. There's going to be high secure platforms in which, in a lot of cases with CDC, when that data comes in with the patient name and a lot of metadata based on the patient, that's put into a very high secure database that has to be protected in a certain way. It has to be validated. And so the technology of the database administrator are going to be the ones that make sure that that PHI is protected, and then also build the conversion engines to de-identify that data so that it can be used for either researchers that don't have that certain clearance, bioinformaticians, where it doesn't matter if the guy's name is Bob, you just want to know, his, you just want to know his blood sugar count, that kind of things. That has to be addressed. Of course, everything I'm talking about is why quality experts are involved, because they're the ones that review the accreditation, the quality accreditation rules, in order to safely and with compliance store the data. And of course, if anything goes wrong here, the management professional is ultimately the one that's to blame. So because they're the one that's leading the project, so they have to make sure that this is all orchestrated in a way that is compliant and can be used, that data can be used to its fullest and its best ability in the future. Big hot topic nowadays, genomic surveillance, right? So one great thing is that they started to take a look at, wow, you can actually detect COVID-19 in wastewater, right? So a community who's, doing their thing, right? In what they found out in cases is that before even the community knows there's a COVID outbreak, before the community has been alerted that there's a COVID outbreak, you can start seeing that outbreak in the waste stream, right? And this is something where you can detect at a community level, but then you're not actually going around and getting people to provide blood every day for a swab sample for COVID. So this is a great technology that's coming into fruition. I'll show you an example about foodborne diseases, about how this is great, but it has also its own challenges. And one of the biggest challenges here with this respect is as you probably know, with very complex sample types, there is a lot of unknown that's in there, I mean, it's just generally understood that we probably only know, when we think about the microbiome, right? We probably only understand a fraction of the percent that's out there, right? So in a lot of cases, what has happened is that, you've taken a look at something, you're looking for something, a target, and your assay may have a false positive because it's detecting something that we've never seen before. And that has been caused a serious problem with some genomic surveillance, particularly when the sample gets highly complex. If you're looking for tuberculosis or you're looking for COVID in a human sample, there's been a lot of technology to, either remove the human sequence either in the wet phase where, it's being removed out of the sample before it goes into the sequencer. In the dry phase, there's also a way that you can basically identify all the human DNA and select it out and take a look at that. That's really well understood. Has that much investigation been done in wastewater? No. So that's something that we definitely have to take a look at. And so in that case, it's the life scientists, it's a lot of times the SMEs on the wet side who understand the complexities of the sample substrate, the background that's in there that is needed, to inform the data scientists like the epidemiologists and statisticians and the bioinformaticians to basically understand what are the limits of the sample collected, if this is collected as part of an assay, if there is a COVID positive, is that COVID positive based on a genomic assay corresponds to actually people getting sick with COVID or not? Those are the concerns, because really still today with public health, actionable public health decisions are still not made simply on a genetic test, right? Because of the false positive and the false negative issues of these things, it really has to be validated by a expressed infection in order to connect these two together to do something actionable for public health. So that leads to actually one of a major project that's part of my portfolio with CDC is the Next Generation Sequencing Quality Initiative. This actually started about 10 years ago. It has exploded. The point of here is to develop quality assurance stage gates, quality assurance rules and policies that make sure that if you're developing a genomic assay to be put into production for use in the public health arena, that it has all of the checks and balances that were done, that the specificity, the reproducibility, the validation against actual disease agents that is occurring. This is really important because, as we all know, over this past decade, genomic sequencing has the speed of genomic sequencing and the cost of genomic sequencing has gotten to a point now where that it can be used widespread in public health surveillance, right? It's cheap enough now. It's robust enough now. It's fast enough that now is within the realm of like the RT, the real-time PCRs that is replacing. So CDC, as well as a lot of public health institutions, are having this mass migration from serology and simple genetic assays to genomic assays. Everybody wants to get these things deployed and out there, but if you don't do the quality checks on it, that can lead to disaster. And there's been incidences at CDC where things like primers for a COVID genomic assay were not quality checked and led to false positive, which then leads to, you know, if you're going to start closing down a military base, or you're going to start closing down a neighborhood or an institution because of that, you got to make sure that you're right. A couple examples that I personally, as a consultant, was asked to by CDC was, and this was like, so back in 2015, there was a college in New York City where the laboratory sent out their grad students and they swabbed everything in a New York subway. They came back, they did metagenomic analysis of that, and they found all kinds of things like the Black Plague. They found anthrax. They found all kinds of things, right? And of course, the news media picked it up. It's wait a second. There's plague in the New York subway. What the heck's going on there, right? There's an example in which, you know, an assay and a bioinformatic process was completed in a public health arena. They said that there was plague in the New York subway, which had caused significant alarms. You know, the Washington Post picked it up. When you actually took and looked at the data, yes, they were finding there are key variants genes and plasmids that are found in Yersinia pestis, which is the plague, that were, the samples came up 98, 99% identical, right? You know, on first, you're like, oh, that's, you know, 99% identical, that's actually, that's pretty good. That must mean there's plague. Well, nobody was getting plague, right? And when you actually took a look at, you know, and this was in the case where, you know, you go to NCBI, you blast your genome sequence, you take a look at what's in there, they find Yersinia pestis. CDC has a collection of Yersinia pestis samples that actually came from people who had the plague. And when you took a look at that sample set, and this was over the span of, I think, of 80 years of people getting the plague, that entire library is 100% identical to the sequence. So what it showed is that 98 and 99% was not sufficient identity to actually connect it to an actual infectious disease agent. And when you looked at, especially these plasmid-borne genes, it was likely, as we all know, plasmids are shared all over the place, right? Horizontal gene transfer going on, and plasmids got antimicrobial resistance, they got metal resistance, they got virulence genes on there. And you looked at some of these examples where the actual virulence genes were knocked out. You can have a 99% identity in nucleotide sequence, but if it leads to a knockout, then that gene, the protein doesn't exist in there. So without proper bioinformatic analysis, they jumped the gun to say that there was plague in the New York subway, which there actually wasn't. Another example here, what's called the BioWatch program. So the BioWatch program is the first line detection of an aerosolized bioterror attack. Okay, so in about 30 to 35 cities across the United States, there are high-powered vacuum cleaners on top of buildings. I know that there's several here in Atlanta. They're sucking air all day, every day onto a paper filter, right? And twice a day, that paper filter is taken out and it's checked for bioterror agents every day. When this program was set up after 9/11. It was set up by the newly formed Department of Homeland Security. They started doing tests, and every once in a while they started getting anthrax, and they started getting tularemia. samples. There was a specific process put in line that if there was a positive result, that reflexed to a point at which you had to start closing down areas. You had to send out the National Guard to do extensive environmental sampling. And what they ended up finding was, it was nice that these assays were highly sensitive, but it was actually picking up what is normal in nature. So you go out to Colorado, probably 30, 40 deer die of anthrax. It's in the, you can pick up a scoop of soil and there's probably anthrax. Here's another thing where, the process was set up, but the bioinformatics and the pipeline and how that was linked to an actionable public health policy was failed. And after they started finding these positives, well, they were positives, but they didn't lead to a bioterror attack. This program had lost a tremendous amount of credibility, which they've never really built.
Pinky, please mute yourself.
Maybe, I don't know. Pinky.
Somebody's mic is open.
Okay.
Pinky's muted now.
Yeah.
Okay.
All right. Yeah, and so in this case here, how the people work together, it takes almost everybody, even the technology experts. You end up with the red box around there. But do you understand how these platforms that are created, and particularly with public health, but it's also in pharmaceutical research, right? It's in commercial, whether or not Coca-Cola is coming up with a new flavor. It takes a village, right? It takes a team to put this together, and how this kind of informs the bioinformatics graduate student is that knowing, keep doing and becoming the subject matter expert of the field that you want to in bioinformatics, but understand at least a little bit to the point where you could at least talk shop with these people, because then you're going to set yourself apart from the workforce, which is going to lead to high success in your life, right? So if you're able to, understand where the scientist is coming from, if you're able to understand what the tuberculosis expert needs in a bioinformatic pipeline, you're gonna develop a successful one on the first try, and it's going to be right, and it's going to be able to get it into the into production and into the public health arena a lot faster. Vice versa, right? I started out as a microbiologist. I dabbled in code a long time ago. I really don't code. I'm definitely not a bioinformatician, but at least I know how to talk bioinformatics shop, right? So like, if my bioinformatic team is trying to develop a pipeline that is to a user interface where a public health official is going to use it, it's got to be, it's got to be right. It's got to be deployed and built right the first time, right? And if you understand at least a little bit about all these different folks, these skill sets, then you're going to go far. Okay, last one here. I think this last one. Genomics surveillance is PulseNet. PulseNet is the CDC's genomic surveillance for foodborne disease. Okay, the reason why it's called PulseNet. Does anybody know what pulse field gel electrophoresis is?
Yeah.
Okay. Just quickly, you isolate the genome from an organism. You put it in a gel. You create a gel. You get bands, right? It's usually two-dimensional with a pulse field gel electrophoresis. And back in the day before genomics, that would be the fingerprint, right? So a bunch of people are getting sick. They find out it's because of foodborne disease. They isolate that. Sample. and they create a fingerprint based on full pulse field gel electrophoresis. That was really important because if you have multiple foodborne outbreaks happening at the same time, you can differentiate between the two strains that are doing it, right? You see the fingerprint of this bug in lettuce, and you see the fingerprint of this one in uncooked red meat. They've done a tremendous job converting it to whole genome sequencing, right? So this is the first example, and one of the first examples of a high quality validated surveillance system that CDC had used for tracking foodborne disease. It was not easy, you know. CDC decided instead of using, they would create their own gold standard reference database, which meant they went back into their freezers and they collected hundreds and hundreds of samples that were directly related to an illness and sequenced those samples. And so that they have a database that knows where every single sequence in that database has been linked to an actual illness, right? Nowadays, if you try to do anything on stuff on NCBI, I mean, there's so much trash in that database, right? That you never know. It's just not usable in a deployed public health asset. They developed very, very specific standard operating procedures. Those procedures were approved by an external accreditation body. You know, if you're going to be part of PulseNet, if you're Arizona Public Health or you're a tribal and you're going to be part of this, you have to do exactly what the SOP does, right? That gets standardization, high quality validation, so that when CDC gets these samples in or gets the data in, they know it is part of a gold standard that they need. And that takes a team, right? That takes a database administrator, that takes a developer, that takes a data architect, that takes a bioinformatician, that takes a laboratory scientist to get to them. One great thing about genomic bioinformatics is the community, this open source community, right? There's tons of open source tools for genomic bioinformatics. And CDC uses those same open source tools. I mean, they're free, right? And they're robust and they work, right? Now, and so like if you set up, one of the problems that they ran into though, is this, you know, they set up, you know, they may have an SOP for sample prep. You know, that may be a specific vendor kit that they say that you use. You know, CDC is an aluminum shop, so most of this is done either on, you know, the MySeq, NextSeq, or HiSeq. You know, and then they developed open source tools to do the assembly. They did develop their own database for PulseNet that ultimately says, you know, this is the specific strain of E. coli that's causing the infection, right? The problem with open source tools is they're being changed all the time, right? Like, you know, that's the one great thing about the democratization of bioinformatic pipelines is that everybody finds new things to do. You know, and it's the same way as foodborne outbreaks happen, those bugs go back into the database, right? Now the problem is that if you have an open source tool that's been changed, in this example here of the assembly going from green to red, that may actually then change the ultimate identification, right? You're moving forward with the idea of creating pipelines that are more specific, more robust, but that lends to a quality issue. So for example, you know, I take a sample, you know, 2 years ago and ran it through this pipeline, and it said it was a specific strain of E. coli. And then I take that sample again and run it now through the updated one, and it's now saying it's a different strain of E. coli, okay? Now, that's a reflection of the modernization and the improvement on the pipeline, but that runs into a quality issue, right? Because now your quality approved pipeline is now producing a different answer, you know, a different answer. And that leads to some quality issues. And so it's really important with respect to the bioinformatician who is collaborating with the IT developer who is monitoring how this pipeline changes. Because you need to have a system in which is called with IT development, a change control system, is that the way for this to work, that you can ultimately keep improving your pipeline, but still maintain a high quality, is that you have to very specifically show the changes that you've made, right? And that is part of a change control system that you put in. So knowing that, like, if I have a sample that I need to test again from 2 years ago, I need the pipeline from 2 years ago to test it for it to be. And so there's a whole part of IT development that is really important in tracking changes as part of a pipeline so that the quality of the development as it moves forward is maintained. And so once again, you know, that's a bioinformatician working with the database guy and the data architect that is monitoring the changes of that pipeline so that they are logged in an archive so that they can be used if needed to go back to in history to do that. And that is very important because you go to a quality accreditation body and that quality manager shows that the pipeline changed and the identification changed, and you don't have that logged, you're going to lose your quality accreditation. All right. So leave my last slide. So hopefully kind of showed you a little bit about what the bioinformatician, its place in this world, its critical place in this world. I think that, you know, a good recommendation with, because I hire a lot of bioinformaticians for CDC, we hire a lot of bioinformaticians right out of the master's degree program. The things, you know, there's this understanding that if you're developing pipelines, a lot of that technology and that understanding is disease agent agnostic, right? If you're developing something for tuberculosis, a lot of that institutional knowledge can be used for any other, you know, HIV. It could be used for anthrax. It could be used for salmonella, right? And so knowing that you're showing successful understanding of developing those pipelines, we need those people right away, right out of school to come and provide that skill set and expertise to our federal clients. But, you know, on a personal note, like when I'm looking at resumes and, you know, nowadays, because of the turnover, looking at stacks of and hiring lots of people, the resumes that rise to the top, that stand out, are the ones where you're a bioinformatician, but you have an understanding, at least an understanding of some of these other components. All right, and with that, I thank you very much, and I'll take a question. Yes.
How on a resume do you show that you know those other aspects?
Yeah, so that's interesting. So nowadays, with the advent of, you know, LinkedIn and Indeed, like all these like huge job and resume aggregating sites, what gets you first picked out is making sure that you have those relevant keywords in the front of your resume, right? Because, you know, our company, as well as a lot of companies, they will use third parties, or they will use basically bots, algorithms to find that big stack of resumes, right? And so having those keywords, you know, if there is a position that is part of the next generation sequencing quality initiative, right? You wanna, and it's a bot, we're looking for a bioinformatician. You want to have all your bioinformatic keywords there, you know, coding, I code in Python and R and that kind of stuff. But, you know, anything of quality, you could relate to quality, make sure that keyword's in, right? That'll get you to the big stack, right? The smaller stacks is like, you know, it's when in reviewing of that stuff is then you're going to want to either have in your resume or amend your resume. So for example, a headhunter company comes to you and says, hey, I found your resume on LinkedIn. It looks like you have the initial set of skills that this job is. We're going to forward your resume to Booz Allen, right? The next thing you want to do is you're going to be like, hey, wait a second, let me get you an updated resume. Because then you want to change that resume to make sure it matches exactly as possible for the job description, right? Because nowadays, because so many resumes that come in, and basically that first and second screening, if the screener or the third party after that initial screen doesn't find one of the qualifications, so you'll see minimal qualifications, nice to have. If you're missing one, your resume goes out the door. So you want to make sure you go back and look at that requisite and you make sure that your resume is addressing every single thing that's on there, you know? And it's not just writing a brand new resume, just develop your resume so that there's a section in your resume that you can easily tweak, you know, like if you're applying to CDC position or Booz Allen or, you know, Bioinformatics, you want to have that place where you're sending what Bioinformatics wants to hear. Or you're sending what CDC wants, what Booz Allen wants. Does that answer your question?
Yeah, thank you.
We have a question from Kelvin Luther in the chat. He asks, working with the military and government, does one have to deal with legacy code in older languages like COBOL? I don't know. COBOL. Okay. Fortran or less common languages like Ada? That's the question.
Yes, unfortunately. I mean, it's becoming less and less, right? But you know, a lot of these federal institutions are dinosaurs, right? They've moved very, very slowly. Now, I mean, what's nice thing, one major aspect of this data modernization funding that I just talked about is that a lot of federal institutions, everything was on-prem, right? You know, that's the way it was built prior to AWS and Azure. But they've been very, very slow to come up with the policy and requirements to migrate those on-prem processes to the cloud or those applications. You know, CDC, they finally figured it out. You know, they're saying, you know, it's like, hey, our preferred is Azure. We're going to require FedRAMP moderate cybersecurity. There's going to be none of these things. So we have a huge amount of work in migrating these legacy systems into the cloud. Part of that is modernizing the code, you know, and that's part of it. It's either, you know, some of it is what's called, you know, lift and shift. You know, you take the existing application from the server, the on-prem server, you package it so that it works in the cloud, and then you just drop it in the cloud. Some cases you got to modernize. You know, you can always just go to ChatGPT and just say, hey, can you convert this COBOL to Python? No, don't do that. I gotta.
That's really.
We can talk about ChatGPT for lunch too. I got a lot of things to talk about.
I think Anne had her hand raised for a bit. So, Anne, do you want to unmute or do you want me to read what's in the chat?
Oh, I'll unmute, I think. Yes.
Okay. You're on mute.
Right. Yeah, the question was about the Million Vets database, and is it as accessible as NHANES, the NHANES database?
Yeah. And they structured it like NHANES, and so essentially it's a little bit more restrictive than NHANES. But what you do is that you request, you go and you request and give them a summary of your project. And I assume it's been a long time since, but I assume that's the same way with NHANES. I know, I think they have some NHANES data that is publicly available already, but they, the Million Vets program, they give it out, but they have this stage gate where you have to request it and provide them like a little project summary.
Well, NHANES also banks certain specimens. Does Million Vets also bank specimens?
Yes, and so, you know, from and I think, you know, you can see on the map, the red triangles are enrollment sites. And so the biorepository for Million Vets program is spread throughout the VA hospital network.
Mm-hmm.
But and I know that they're trying to consolidate it into Jamaica Plains, which is a VA center up outside Boston.
Yeah.
So yes, they are. It's kind of spread out. As of right now, I don't know if you have that you can gain access to the samples themselves. It may
NHANES will sell you the specimens. I don't know whether that's the case. I mean, generally they're sold to people who have approved proposals. So yeah, thank you very much for that. We can talk about that later too.
Yeah, sure. Yes.
So I know there's some restrictions on federal employment based on citizen status. As a consultant for federal government or like contractors, is that a little more flexible based on citizenship and other aspects?
Yeah. So there, you know, there's a bunch more hoops that you have to jump through than just the standard, getting a visa and, but, I've had two colleagues of mine who have gone through the entire process who have become citizens. And a lot of these companies will support and fund your progression. So they'll make sure, like Booz Allen, we have an office that manages and monitors all of the people that have different types of visas or going through the green card process. Booz Allen will and other companies will pay for the legal fees and the legal support. There are some restrictions. I know somebody who did Air Force Research Labs. She's British and she's now a US citizen. And so at that point, she was able to go through the entire process, but also have a security clearance. In HHS, the more like public health, it's a bit less restrictive than the military, but there's definitely, you got more hoops to jump through, but it's definitely doable.
Thanks.
Okay, I think we can do one last very short answer to a very complicated question from Oliver in the chat. Oliver asked, what reassurances can you get as a bioinformatician about how your work will be used? If genetic risk factors for hearing loss are identified, being denied coverage for it because they are already predisposed, or service members being denied protective equipment if they don't have the risk factor, can these concerns be addressed while you're also job hunting?
Addressed job hunting. I will say that I have yet to see a negative, and this is just in my personal sphere, it may exist or not, but, I have not seen projects in which we have linked genomic predispositions, to specific disorders, and that being seen and actively used to block or inhibit, like in the military and that stuff. The unfortunate thing, it probably exists. It's more of the military is, and this is the way that, you know, I see it as my opinion, is that the military wants to support and fund healthcare for these really complex problems like PTSD. You know, one of the biggest ones is the respiratory issues that soldiers have in their late years that are, everybody understands, are attributed to the burn sites and forward deployed bases, but there's no direct validation between the two, right? And so I've been hired and my teams have been hired to find those connections. And if you find it with the assumption that then the military says, oh yes, we do see a direct connection for those soldiers who were close to a burn pit, who have this weird respiratory disorder later in life, they want to do that connection so that then that validates spending healthcare money towards it. Maybe my opinion is a little bit idealistic. I'm not quite sure, but it seems like that's the way that they're looking for. I think, is that the question? Do you know?
I think so, but we're out of time anyway, so that's all we're gonna get. All right. Thanks, everyone.
Whether you like it or not.
Oliver says yeah.
Oliver says
What do you want?
yes, I may. Oliver's happy enough.
No, happy enough.
All right, let's
He's not rocketing in that matter.